Good morning, and thank you all for joining the New York Botanical Garden as we kick off We Dig Bio 2021. My name is Charles Zimmerman, and I am so pleased to welcome everyone who's joined in today to learn about historic plant collections at NYBG and the important ways that community scientists support our work to document and conserve the plant kingdom. At any time during today's event, please feel welcome to submit questions by clicking the Q&A button at the bottom of your Zoom window, and our hosts will raise these for our speaker to answer at the end. Live captions can be enabled by clicking the CC button, then show subtitles or view full transcript. This presentation is being recorded, and it will be shared for later viewing on the NYBG Lecture Library. Worldwide engagement for digitizing biocollections, or We Dig Bio, is a semi-annual 4-day event led by museums, universities, herbaria, and other organizations working to preserve and share natural history specimens with researchers and the public. Our common aim is to mobilize community scientists to help create digital data for biodiversity collections and support their extended use for understanding nature. Over the next 4 days, thousands of individuals around the world will join together for online workshops, collection tours, research talks, and citizen science expeditions. You can learn about all the great events happening by checking out WeDigBio.org or follow @WeDigBio on Twitter. This morning's event marks the beginning of a 4-part We Dig Bio series brought to you by the New York Botanical Garden. Whether you're an aspiring citizen scientist, a longtime volunteer looking to connect with others, or if you're simply interested to learn more about the science of plants, there's an event that's right for you. There's still time to register, so check out the link on screen and in the chat to learn more. Now it is my great pleasure to introduce Nicole Tarnowski, Assistant Director of the William and Linda Steer Herbarium, who's here to lead us on a virtual tour of NYBG's historic plant collection. Nicole, I'll turn things over to you.
Hi, good morning. I'm happy to be here with you today, and I thought we would start with a little video meant as kind of an intro about the herbarium.
Hello, and welcome to the Herbarium. I'm Nicole Tarnowski, Assistant Director of the Herbarium, and I'm excited to give you a behind-the-scenes look inside the Herbarium of the New York Botanical Garden. Our Herbarium is the largest in the Western Hemisphere, holding approximately 7.8 million specimens, documenting the plants of the world over the last 300 years. The collection is housed in a state-of-the-art, climate-controlled facility within cabinets designed specifically for herbarium specimens. In fact, this design dates back to the collections of Carl Linnaeus, who created the system of taxonomy that scientists still use today to name and categorize natural history collections. The herbarium is curated with a meticulous filing system organized both by the scientific name, the family, genus, and species, as well as by the geography of where the plant was growing when it was collected and preserved. Each specimen consists of the physical plant, which has been pressed and dried, much the way one would press a flower in a book. There is also a label to tell you what the specimen is, its scientific name, exactly where it was collected, including latitude and longitude, which allows us to map it. The label tells you who collected it and when it was collected. This information is as important as the specimen itself. Preserving herbarium specimens has been done using the same process for close to 500 years. It is still the best way to preserve plants for study. The specimen is dried and pressed in the field using a traditional plant press. The specimen is arranged in absorbent paper such as newspaper, making sure to show all of the most important structures needed to identify it. It is sandwiched with corrugated cardboard, a wooden frame, and tightened with belts. Once the specimen is dry, it is sent to the herbarium where we can do the final step in the preservation process, mounting it onto a sheet of paper so that it is supported underneath and can be handled as it is studied. Our mounters will first arrange it on the sheet, being sure the important structures are visible, but also with an eye for design. Obviously, these specimens are created for scientific use, but there is also a beautiful art to preserving and presenting the specimens in this manner. Every specimen gets a small packet so that we can save any pieces that may fall off. These fragments are, in fact, very useful to researchers as they can extract DNA from them. Recently, one of our specimens, originally collected in 1829, was sampled and DNA was able to be extracted. We then drizzle glue on the back of the specimen and attach it to the paper. The glue is a water-based white glue that is similar to Elmer's, and it is acid-free and archival quality. While the glue dries, we weight down the specimens and cover with wax paper. To reinforce gluing them to the sheets, many specimens are also secured by sewing them to the paper. In contrast to the traditional practice of mounting herbarium specimens, these days we also focus our efforts on digitizing our specimens so that we can share them online with both researchers and the public. Our digital imaging lab contains equipment specifically designed for herbarium specimens. We use high-resolution instant capture cameras and light boxes, which allow us to provide diffuse lighting for each specimen image without needing to use flashes. The image is taken and checked on screen. You can see the level of detail in our images by zooming in to see the many important structures of a specimen. The herbarium contains many treasures and its contents tell many stories. I'm looking forward to telling you more.
Great, so I hope you enjoyed that little intro to what a herbarium is, and I am looking forward to telling you more. I thought we would start with a little bit of history about herbaria in general, with the first known herbaria, which was created by a physician in Italy in the late 16th century named Luca Ghini. And he preserved plant specimens almost identically to the way that we do it today. He pressed and dried them and attached them to paper. You can see from the photo on the left there that he actually bound these into books, which is different than what we do now. But his purpose for collecting and preserving these specimens was to use them as a reference collection of medicinal plants. And Luca Ghini made studying medicinal plants an essential part of medical training for his students. And all of his students continued this tradition throughout Europe. So moving forward through the centuries, by the 18th century, there were many botanists creating this more as a formal study of studying plants into the study of botany. Where botanists are describing and categorizing and naming different plants. So in the 18th century, Carl Linnaeus, who is considered the father of taxonomy, published Philosophia Botanica, where he advocated that instead of binding these collections into books, that each specimen should be mounted on a separate sheet of paper. And the idea behind this is that in categorizing different groups of plants, your ideas are constantly evolving and developing, and he wanted to be able to reflect that in the way that he housed his specimens and filed them. So if each specimen was on a separate sheet of paper, it meant you could reshuffle them to reflect the current ideas of and understanding of those plant groups. So you can see the photo on the right is a drawing of Linnaeus's actual herbarium cabinet where he stored his own herbarium specimens. And the dimensions of these cabinets are almost identical to what we use even today to store our specimens in the herbarium. Around the same time, there were many botanists by this time in Europe that were interested in studying plants, not just in their own locality, but in other places in the world. This was, of course, also the age of colonialism. So many European countries were launching colonial expeditions to go to all different parts of the world. And quite often there were naturalists on those expeditions. The British expedition led by Captain Cook included the botanists Joseph Banks and Daniel Solander. They traveled the world with Cook on this first expedition here. This is a specimen collected on that expedition in 1770, I want to say, in New Zealand. And this is actually a ranunculus species that was considered new to Western science. Obviously, the indigenous population in New Zealand probably knew about this plant already, but from a European standpoint, this was a brand new species. And so this is one of the older specimens we house in our collection. Other illustrious collectors that we have represented in our collection include Charles Darwin. We have several specimens collected by Darwin. This one collected on his voyage of the Beagle in the 1830s. If you think about it, obviously these botanists have made journeys to faraway places and brought these specimens back. And once the specimens themselves are housed in the herbarium, they sort of continue that journey. And you'll notice that every annotation and stamp and illustration on every sheet is another, reflects another use of the specimen by a researcher. Every researcher that looks at them will put their own opinion or something on the sheet. And so that's all reflected in the journey of this particular specimen. Turning our attention more to the Americas, at the same time, there were quite a few American botanists in the 19th century. One of the first public botanic, or the first public botanic garden in the country was Elgin Garden. This was in New York City. It's actually the footprint of where Rockefeller Center stands today. It was founded by Dr. David Hosack, so continuing that tradition of physicians really making botany an essential part of their medical training. David Hosack did his medical training in Britain and became extremely interested in botany. And when he returned to New York with his own funds, established this botanical garden. It included 2 greenhouses and a herbarium, and he grew medicinal plants, he grew horticultural plants, and some native species as well. This is an example of one of his herbarium specimens. And unfortunately, because this was done with his own funds, he wasn't able to keep it going for more than about 10 years. The property was eventually ceded to New York City, and it eventually landed in the ownership of Columbia College, which at the time had still been in Lower Manhattan. So they moved their campus up to 49th Street, where they were for much of the 19th century, while the entire grid of New York City sort of grew up around them. So if you think about what a wide, a large space Rockefeller Center is, it's partially because that was first a botanic garden and then Columbia University's campus. And so the grid of New York City didn't run through it. In the 19th century, another very prominent botanist, American botanist, was John Torrey. He was based in New York. He was extremely prominent throughout the entire country. He was basically in touch with every botanist throughout the whole country that were also going on expeditions, studying westward expansion and railroads. Every one of those expeditions also had botanists on them. They were all sending specimens back to John Torrey. By the end of his career, he had amassed a personal herbarium of about 50,000 specimens, which he donated to Columbia College. He also, towards the end of his career, established the Torrey Botanical Society. And this was this is still in existence today. They just recently celebrated their 150-year anniversary. It's still supported here at the Botanical Garden. And this was a society that was available for both student and amateur botanists to get together, have discussions about local plants. They would often go on little local expeditions to various places around the city. The photo on the right is the Tory Club taking an expedition up to the Bronx, taking the train up to Pelham Station. And so this was a really wonderful resource just for anyone, and continues to be a wonderful resource for anyone that's interested in plants in the tri-state area. Which brings us to the founders of the garden, Nathaniel Lord Britton and his wife, Elizabeth Knight Britton. They were both botanists. Nathaniel Lord Britton enrolled at Columbia University just 2 years after John Torrey passed away. Elizabeth Knight Britton was also a biologist. She was a student at the Normal School, which is now Hunter College. And they both, separately developed this passion for plants, and both became part of the Torrey Botanical Society. They both went on those same Torrey expeditions, some of which were before they were even married. I like to think that part of their courtship was going on these expeditions and botanizing together. These are two specimens that they collected together before they were married. The one on the left was collected in the Kingsbridge section of the Bronx in May of 1883. Nathaniel at one point remarked on visiting the Bronx, "It was this trip that gave me my first knowledge of the natural beauties of the Bronx Valley." So they were married, and they took their honeymoon to London and visited Kew Gardens, which at the time had already been a very well-established botanic garden with amazing facilities for botanical research, including living collections that reflected, a lot of those colonial expeditions that brought back plants from all over the world that they continued to propagate in the greenhouses. And they had an extensive herbarium collection that was more extensive than anything that existed in America. So many American botanists really needed access to that type of collection. And so the Brittons were incredibly impressed with Kew. And when they returned from their honeymoon, they came back to the Torrey Botanical Club and really lobbied to establish a botanical garden here in the US. So the Torrey Botanical Club published this public appeal, and they were able to garner the support of many of the major philanthropists at the time, including Andrew Carnegie, J.P. Morgan, Cornelius Vanderbilt, who all ended up being on the original board of the garden. And so we were incorporated in 1891. And this photo on the right is actually the Torrey Botanical Club taking hold of this property for the very first time. The hillside that they're all walking up is actually where the Tulip Tree LA stands today. So, much to many of those founders and all of the hard work that went into establishing this institution, that all continues today. And, over 125 years later, we really have grown into the premier research institution that the Brittons and even David Hosack had envisioned for having a botanical garden in America. Our scientists today collect specimens and do research in many corners of the world, collaborating with botanists in many corners of the world, bringing herbarium specimens back here to our collection, which now numbers about 7.8 million specimens. We're one of the largest in the world. And of course, these specimens each one of them is extremely valuable for research. Every single specimen is really a physical voucher that represents that species collected in that place at that particular time. So and those are three important pieces of information when a researcher is doing botanical work. The first thing that needs to be done is to identify the specimen, right? So unless you really know what the species is, you can't really, it's not as valuable. So some of you may be familiar with even popular field guides of, say, wildflowers, where they're often organized first by flower color, and then you sort of narrow it down from there. And scientists use something similar when they're identifying specimens. We use dichotomous keys, which use many detailed characters of specimens that sort of lead you through these questions and lead you to the ultimate answer of what the species is. Once you know what that name is, there's many questions that can be asked and answered about that species. Many researchers, as part of their career, become a specialist in a particular plant group. Someone might spend their entire career just working out in what we call monographic work. They will detail every species in a plant group with, extremely detailed species concepts. And they might do some systematic work to really figure out how all of those species are related and where they fall on that family tree. Another important aspect of herbarium specimens is the geography for where that plant was growing in the world. And because we have such an extensive collection, we can easily have hundreds of specimens of the same species. And if you pinpointed each one of those specimens on a map, you would start to get an idea of the entire distribution of that species, right? So you can kind of understand the boundaries of where that species exist. And this is really important information that our citizen scientists are helping us to extract out of these specimens and record. Another interesting aspect of the geography on specimens is that in addition to specialists, in addition to researchers being specialists on a particular plant group, they might also be a specialist in a particular geography, right? So if you're studying one species in all the places it grows, you could also study one place and all the different species that grow there. A third really important aspect of the data on specimens that, again, citizen scientists are helping us to record is the exact point in time when that specimen was collected. And this information, if you think about it, we have specimens going back hundreds of years at this point. We have a record of what has been growing in the landscape and how that landscape has changed over time. So our specimens are increasingly used for climate change studies, including phenology studies. So the study of phenology is tracking the life cycle of an organism, right? So for plants, you can track exactly when something has flowered, when it has fruited, when the leaves fall off in the fall. And so using our herbarium specimens, you can go back to much earlier specimens, see exactly when something flowered. In this case, you can see the specimen on the right was collected in 1891 on June 16th. You jump 80 years forward, that middle specimen was collected in 1972, a week earlier on June, 12th or 8th. And then if you jump another 30 years forward, that specimen on the right was collected in 2011, many weeks earlier on May 26th. So, and robust phenology studies are using many specimens and getting, obviously there's change on a year-to-year basis, but you can also extract it using much more data to really start to see these trends of spring warming up sooner every year. Another way of studying how the landscape changes is by studying invasive species. And again, our specimens go back many hundreds of years. So we can track when invasives first became established in a particular place. If anyone is familiar with purple loosestrife, it's a very pretty plant with these pretty purple flowers. It's originally from Europe, and it has become an invasive species throughout most of North America. This particular specimen that we hold in our herbarium is considered one of the very first herbarium specimens documenting that purple loosestrife was now growing in nature here in the US. And so this specimen was collected in New Bedford, Massachusetts in 1831. So that specimen is sort of ground zero for purple loosestrife getting a foothold in North America and then spreading. You can see these maps on the left are another study that was done of purple loosestrife growing in the Great Lakes region. And again, each one of those points represents a specimen, and you can see throughout time how there were more and more specimens being represented as this really took a foothold. Our collections can also represent rare species. This is a trillium that grows in the state of Georgia. And species can be rare for a variety of reasons, one of which being that they might just have a really narrow distribution, so we call them endemics. If something is endemic just to one small area, it is just intrinsically a rare species, as is the case with this trillium. We also, unfortunately, have extinct species represented in our collections in the herbarium. One of our interns a year or so ago back went and combed through our entire herbarium with what's called the IUCN Red List. And this is a list of every known extinct species in the world. And so she was able to go through that list, and she found, I think, about 28 specimens of species that we have in our collections that no longer grow on Earth. Another interesting use of herbarium specimens is studying wild relatives of crops. It should be noted that the majority of collections in the herbarium are plants that have been growing in nature. They're not plants that were cultivated, right? But that does include relatives of many cultivated species. So these are two specimens. The one on the right is teosinte, which is a wild relative of maize, and the one on the left is domesticated maize or corn. And this is an interesting use of herbarium specimens as well. If you can narrow down traits in some of the wild relatives of crops, sometimes you are able to find traits that might be better suited to our changing environment. You can find traits that might be more drought or heat resistant. You could also find traits, if it's necessary, of flood resistant specimens. So this is a really interesting use of studying some of the wild relatives and some of the additional diversity that's out in nature still that we can possibly help in our cultivated plants. Molecular biology has also been a huge use of herbarium specimens. You can actually take a small sample off of a herbarium specimen and extract DNA out of it. And these types of molecular biology studies have many different uses. One can isolate specific genes that are responsible for various traits in a particular plant. This particular study was done by some collaborators of ours from the University of Florida, who came up here, sampled thousands of our specimens, and they were studying how different species that are capable of nitrogen fixation, and they were isolating some of the genes that are responsible for that nitrogen fixation. So you may or may not know, there are some plants, many legumes, that are actually able to grab nitrogen out of the atmosphere and incorporate it into their plant tissue in their own metabolism. And then there are many plants that are not capable of doing that, and many of our crops that are not capable of doing that require a lot of fertilizer. So this would be a really interesting use if you can, if they can isolate these genes that are responsible for nitrogen fixation and use them in other crops. That could really be useful in cutting down the use of fertilizer. In addition to some of these historical stories that I've told you about, and many scientific uses of herbarium specimens, I personally just think they are works of art. The entire process of pressing them and gluing them and sewing them down onto paper, doing these beautiful layouts, I think is a beautiful process. Each specimen is sort of its own individual portrait, right? It's been removed from the landscape so that you can really focus just on that one species. And the skills themselves are skills that really have been cultivated just in herbarium staff. They're passed down through the generations of herbarium staff, though we do currently have a very popular continuing ed class here at the New York Botanical Garden that's partially taught by one of our mounters, if you're interested in learning how to mount specimens. But it is really just a beautiful art and a craft. So in conclusion, I'd just like to say the herbarium is a museum of sorts. It is, 7.8 million specimens all in one building, and it represents what grew in nature in the past and what is growing there now. It is, of course, archival, and with the right stewardship, as we hope to do, these specimens are going to be available for future generations of botanists to study. And so with that, I thank you, and I guess we'll open it up to some questions.
Thank you so much, Nicole. That was terrific. I really enjoyed that, and so did our audience members. We have a lot of great questions, and it's time now for the Q&A. So audience members, please go ahead and add your questions to the Q&A at the bottom of your Zoom screen if you haven't already. And we also invite you to upvote the questions that others have already submitted, and that'll help us identify the questions that are most interesting to all of you. So we've got lots of great questions. Nicole, I'll just get us started with one of them that was asked earlier on in the presentation. You mentioned a little bit how some specimens arrive at the garden being collected by NYBG scientists. Could you tell us a little bit more about how collections make their way to NYBG, both internationally and in the United States?
Yeah, absolutely. All of our curators that go out collecting in whatever part of the world they are in generally have collaborators in that country. So they're working with local botanists and often are going out collecting together. When someone collects a specimen, they actually will collect several duplicates of that same plant, right? There might be 5 or 6 duplicates of that same, if it's a tree, 5 or 6 duplicates of that same tree. They're all considered that same collection. And that allows us to make sure to leave at least one duplicate, sometimes more, in the host country where they should be left for local researchers to study. It allows us to take some back to the New York Botanical Garden here. We can keep one for our own collection, and sometimes we have additional duplicates that we can share with other institutions as well. So every herbarium is doing this. We're all, with any additional duplicates that we have, we're all sending them to each other, and that really benefits, not just one institution, but many.
Terrific, thank you. Several of our audience members were curious how color is recorded on specimens.
Yeah, it really depends on the plant. Some of them hold their color really well. Some just turn brown. If you think about tropical, lush plants, you think they're going to be these, beautiful things, but often because they have so much water in them, when they're pressed, they turn brown. But we do record another piece of information that's recorded on the label that I didn't speak about is there is a plant description. And so someone might say the colors are yellow, the flowers are yellow, the bracts are green. So it's really more of a descriptive process in terms of recording the colors. We do, on some specimens that we have digitized, include a color chart that can be calibrated if someone is doing some sort of study comparing the colors on various herbarium specimens.
Excellent. Here's a 2-part question about how the herbarium is organized. So firstly, can you tell us a little bit about that? Is it based on taxonomy, geography, a combination?
That's right, it is a combination. It's first and foremost organized taxonomically. We have a list of all of the plant families that we recognize. We actually assign each family a number. And so that allows us to put families that are closely related near each other in the herbarium. So if someone is studying one plant family, but maybe their sister group would be filed in the next cabinet over. Our herbarium is 4 floors. So for someone that needs to study related plant groups, it's really useful to have them next to each other. So it's first organized by taxonomy, and then there is within that things are separated geographically, because that, again, is another important piece of information of where that plant was growing. So those two pieces of information really reflect how researchers are using these specimens.
Terrific. And this is the next part to that question from Lauren. So in your filing, how do you deal with name changes and rearrangements of family and genera?
Yeah, it's a lot of work to keep the herbarium organized. We have a wonderful staff here that tries to keep up to date with, if you're familiar with taxonomy at all, that names, as researchers learn more information about a particular plant group, they're constantly, just like Linnaeus, are constantly revising their idea of how things are related. So that is reflected in name changes constantly. Which means we then have to go back into the herbarium and move, sometimes hundreds of specimens from one species name to another. It is a challenge to keep up with all of this. We also have a mechanism of keeping track of this in our internal database. So if the specimens themselves, if we haven't had a chance to move them, that we also record that information in our database.
Thank you. Next question comes from Patricia. How can we submit specimens to the Steert Herbarium? I think we have a collector on the line.
Hey, we generally, we do have sort of a set of standards for what makes a good specimen. They have to be well pressed and well preserved. We hope that when they're collected, they are fertile. So that means they are either in fruit or flower, or both. They also have to have really good data that comes along with them, right? So that label information has to be really extensive. Generally, most of our collectors come from the sort of the academic community of botany, right? So we've got our own researchers and all of their grad students who are really learning this process of how to collect things scientifically, because we also don't want to collect just every single weed that is everywhere. We want to sort of target the really important plants that need to be documented. So those are all some parameters.
We have a question here about preservation. Does the New York Botanical Garden use any kind of poison for helping to preserve specimens?
That's the neat thing about this process, is that once the plants are pressed and dried and then, just attached to a sheet of paper so that they don't disintegrate, they don't need any other chemicals to preserve them. Yeah, it's a pretty simple and straightforward process that, hundreds of years later is still the best way to preserve them. We do, one worry in a herbarium is insect infestation. There are a few types of beetles that love to eat dried plants, and they can completely destroy a collection. And so to preserve them and protect them, that's why we keep them in herbarium cabinets with closed doors so the bugs can't get in. And we also freeze the specimens before they're brought into the herbarium to kill any insects that might be hidden somewhere in the specimens. But in terms of chemicals, we don't have to use anything.
Which is fortunate for all of us herbarium staff. Next up, can extinct plants be regenerated?
There has been some studies to try and germinate seeds that are on herbarium specimens. It really is going to depend on that particular species. There's some seeds that are able to lie dormant for many, many years. Others aren't. So really, it would depend on the particular species. There have been a few studies of trying to germinate seeds specifically from herbarium specimens. It was a pretty low percentage, but they were able to get at least some activity. So it might be possible. I won't say it's very likely, but it is possible.
Terrific. We have a mycologist in the audience. Christina wants to know about whether our herbarium also collects mushrooms. And if so, do we collect the mycelium or just the fruiting bodies?
I think we collect both macro and micro fungi. We have a pretty extensive mycological collection here, actually.
Awesome. Oh, this is a great question. Do collectors record fragrances as well as colors?
Sometimes they do, yes. Yeah, right. So sometimes, for those of you that are citizen scientists and have been reading some of our herbarium labels, you might come across something in the plant description that not only describes what the plant looks like, but sometimes they do describe the fragrance. Sometimes they'll describe insects that might be visiting them as pollinators. So there is quite a bit of additional information about particular collections.
Thank you. Thank you, Nicole, for answering all these questions. We've got so many good ones coming in. I'm trying to get as many as we can. Um, do researchers examine specimens to study speciation and past geographical separations and the relationships between these related species on different continents?
Yeah, absolutely. I mean, one of the greatest strengths of a herbarium is that we have the benefit of, hundreds of years of collecting, and a large herbarium like ours has many examples of the same species. And if you're familiar with what a species concept means, there's variation within a species. So not every single plant is going to have exactly the same traits. And so having a large number of specimens of one species means you can compare all of those variety of traits that are still considered that same species. And again, because we have that geographical information, you can place that plant in that place and then over time.
Terrific. I think we have time for two more questions.
Great.
We'll have to cut it off there. This question comes from Patricia. How can we study a specimen that it is in your collection, but it has not yet been digitized?
We do offer a service for our researchers. If someone really needs a specimen that has not yet been digitized, we do offer a service called an e-loan, where we will go into the herbarium and pull any specimens that are needed, and we will digitize them as a e-loan to a particular person.
But it certainly does help when we do have them digitized. So we are working as hard as we can to get as much information online as we can. And all of you who participate in our citizen science expeditions make a huge impact on making that possible. So last question comes from Carmen. Love this question. I hope you will too. If you could design the perfect herbarium, how would you want it to be? And I'm referring to particular storage and environmental variables and access.
That's a great question. I think we're very fortunate here at the New York Botanical Garden to have a really nice facility that was designed with the best practices in mind for keeping our specimens safe, and also for ease of use for our researchers. So our own herbarium is climate controlled. It's both temperature and humidity controlled. So we keep it at a fairly low temperature so just in case any insects are able to get in, hopefully it's cold enough that they can't reproduce. We also keep it at 50% humidity to prevent anything from ever molding. If you think about it, these plants are all glued on paper, so we don't want anything to have a high humidity that could get moldy. Our herbarium in particular, we have these great counters that are about 10 feet long. And so, often a researcher might come, they might have to compare 200 specimens, and we have these nice long counters where you can really spread out and really sort through your specimens and have the space to do it. Those are some of the things I like about our herbarium that have been incredibly useful.
Terrific. Nicole, thank you so much for taking the time to help answer all these questions and teach us all about the herbarium and give us all these great historical stories about where collections are coming from and how they're being used. We really appreciate it.
Great.
Awesome. So we have a couple more questions, but unfortunately we are out of time, everybody. Thank you so much for being here, everyone in our audience for joining today's presentation. If you missed any part of the event or wish to watch it again, a recording will be saved and shared online on the NYPG lecture library. So We Dig Bio has only just begun, and I look forward to seeing many of you this afternoon and tomorrow as we continue learning about biodiversity collections, their use for research, and how citizen scientists around the world are working to unlock them. So you can learn more and register for all these upcoming events in the link in the screen share and in the chat. Don't forget that We Dig Bio lasts until Sunday, April 11th, and we hope you'll spend time before then getting involved in NYPG virtual herbarium expeditions. Your participation makes a tremendous difference for scientists around the world, and we're so grateful for your enthusiasm and your dedicated support. Until next time, please stay well, stay green, and take care, everyone. Goodbye.